Okay, so here we are. I first just want to go over and do the basics of what DNA structure is, right? So when you look at this, here is a really cartoon version of what DNA double helix looks like. So here over on this side, it's all twisted up, and we're just going to take this section and blow this up and look at it, right? So as you can see, once again, these phosphates, right, they're all the same molecule. And over here, you have the exact same thing. These phosphates, they are all the same molecule. These guys right here, the D's, they're actually sugars. Okay, these sugars are what we call a deoxyribose sugar. And when it comes to the sugar, there's lots of different sugars that are out there. Remember, you've got deoxyribose, which is what's in DNA. You got sucrose and fructose and lactose, all in with ose. So these are all sorts of different types of sugars, right? Hence why DNA has the name deoxyribonucleic acid, because it's got a deoxyribose sugar. So here's the sugars. Here are the phosphates. They are the exact same on this side as they are on this side. Okay? So this is what we call the DNA backbone, because the backbone never changes. It stays the same. It's strong and it's sturdy. It doesn't change. Now this part right here, these are your nitrogenous bases, and they can change. But as you can see, there's 4 different types of them. You have A's, which stands for adenine. You have T's, that stand for thymine. And you come down here and you have C's, that stand for cytosine. And G's, that stand for guanine. So here you've got your DNA double helix, right? You've got the sugar phosphate backbone. That's the exact same on both sides. Then you have your nitrogenous bases in the middle. Now, you might have noticed these dotted lines that are between the A's and T's and the C's and G's. And those dotted lines, they are what we call hydrogen bonds. And these hydrogen bonds, they're weak. And it's mainly because we're going to break this puppy apart. And so we want these bonds to be weak. So these are weak hydrogen bonds. Now, these lines right here, they're not broken up. Okay? These bonds right here, they are solid, rock solid. So they're a different type of bond. Okay, these bonds are called covalent bonds, and they're strong, and they don't like to be broken apart, okay? Which is a good thing. We don't want them to be broken apart. So between the nitrogenous bases, okay, between the bases, we have weak bonds, weak hydrogen bonds, because we're going to break this puppy apart. But on the sides of the DNA double helix, the backbone, they're strong covalent bonds. We don't want them to break apart. All right, so there's a couple more things I want to point out. Is that, like we said, DNA is made up of nucleotides. And a nucleotide is made up of 3 parts: a phosphate, a sugar, and a base. And that's it. So DNA in general is just made up of a bunch of nucleotides. Okay? So you got a phosphate, sugar, base. There's 1 nucleotide. Phosphate, sugar, base, 2 nucleotides. Phosphate, sugar, base, 3. Okay? And then, oh, you don't have 1 here, so that's not a complete nucleotide. And we come over to this side. Phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. Okay? So there is your DNA double helix in a nutshell. Okay? So we're going to go ahead and go over to our notes packet on page 4, and I want to color code the DNA structure. So I have a picture of one right here. This is what you're going to be doing in your notes packet. Now, I know the picture's a little faded, and so that's why this individual on the backbone kind of rewrote over what the letters are. But just keep in mind, if this is a phosphate, the next one is a sugar. So you have a P for phosphate and the D for a deoxyribose sugar. So that's how it goes: P and then D, and P and then D. Okay, and it just alternates the whole way down. Okay, same thing over here. Alternates the whole way down. Now you'll notice here, this one's a phosphate. Well, over here, this one's a deoxyribose. So it goes D, P, and alternates the whole way down. Okay? So we are going to color code that. Now I'm going to go ahead and jump over to our notes packet. Scroll down. So here's where you're at. So I need you to choose, when it comes to your colors, you're going to need a color for your phosphate, a color for your sugars, and a color for your hydrogen bonds. So there's 3 colors right there. As far as your covalent bonds, you do not need to have a color there. You can actually just leave that white. But you will need a color for your A's, G's, T's, and C's. So we're looking at 7 different colors. So go grab those colors. All the colored pencils are in the back of the classroom, and they're on the lab tables. So you can use markers, anything you would like that's got color, but you need to pick 7 different colors. Okay, I hope you got your 7 different colors. So first off, let's go ahead and choose whatever color you want for your phosphate. And then you need to pick this one that's lettered P for phosphate. And then you're going to alternate all the way down, like I showed you before. And then same thing, pick another color for your sugar. Start here with the deoxyribose, your sugar, and alternate all the way down. I'm going to go ahead and flip over to a student example that I have here. Sorry, it's a little bit blurry, but you can get the idea. So there's their colors they chose. You got your phosphate, your sugar, the hydrogen bonds, in case you're wondering, well, where are the hydrogen bonds? So when it comes to the hydrogen bonds, so you've got their phosphate sugars, phosphate sugars, phosphate sugars. Well, your hydrogen bonds, remember, are between your nitrogenous bases, and they're weak. So the H represents a hydrogen bond, so they colored hydrogen bonds all the way down their whole picture, right? Now your covalent bonds, you don't need to worry about, like I said, color coding covalent bonds. We know that the covalent bonds are right in here between your sugars and your phosphates. Okay. All right. Next color you'll want to make sure that you pick and choose would be those nitrogenous bases. All right, your A's, G's, and T's, and C's. And just make sure that when you color code them, it tells you the letters of what things are. So you'll pick your color, G, and then color the G all the same color all the way down. Same with your C's all the way down, and your A's and T's. Okay. All right, so let's go ahead and get this part color coded, and then we'll meet back up. All right, so I want to have you circle a nucleotide. Okay? So this DNA double helix is full of nucleotides. There's lots of them. But we're going to go ahead and circle one of these nucleotides. Remember, a nucleotide is what makes up your DNA. And a nucleotide is made up of 3 parts. Right? It's made up of a phosphate, a sugar, and a nitrogenous base. Okay? And let's go ahead and label that being a nucleotide. All right, so there's my labeled nucleotide, my DNA double helix. So you got this whole thing colored. And now we're going to go ahead and move down to the bottom part of the page, which is taking some DNA structure notes. Now I'm going to make a recommendation that for these DNA structure notes, you will probably want to have your own sheet of paper so you can add, so you can just use as much space as you want to take these notes. So you're going to put in notes here. And then, like I said, you might want to have a separate sheet of paper to take some additional notes. All right, so what I want you to put down where it says DNA structure notes is, let's go ahead and talk about it. All right, so first off, DNA. It is a nucleic acid. All right, so it is a type of nucleic acid, and it's more particularly, like we said, a deoxyribonucleic acid. It stores information. That's its job. Okay, DNA is to store information. And it stores information so it can make those proteins. And it stores information to pass on traits from one generation to the next. So your DNA is a genetic code that we have that we can pass on to our children. Right? But the purpose of DNA is to make proteins so that your body can function. So this is going to go in that space where it has DNA structure. So you can go and just pause the video and get this written down as needed. All right, so we're going to go to the next slide. So what is DNA like? And like we've seen from the videos, we know that DNA is a chain-like molecule, and the chain-like molecule is made up of nucleotides. So when it comes to what your nucleotide is, we know that your nucleotide is made up of those three molecules, a phosphate, a sugar, and the base. And that's what forms your double helix. So what does your double helix look like? It is two individual strands all twisted together right here. So here's a good example. There's one DNA strand, that's your sugar phosphate backbone, and then this right here is what your nitrogenous bases are. So that's one DNA strand, and then you have the complementary DNA strand right here, also made up of a sugar phosphate backbone, and then you have the other nitrogenous bases that pair up with it. Now this DNA is going to coil up nice and tight, like from the video we showed you earlier, into chromosomes. DNA has a certain unique direction. And that's where you're going to hear things later on about 3 and 5 prime. And I'll draw that up so you can see what that looks like. Alright, so let's keep rolling into what a nucleotide is. So do me a favor, and once again, under DNA structure, I want you to write this down. Sugar, phosphate, base. And then we are going to draw us a picture. So DNA is made up of nucleotides, and it's a sugar, phosphate, and a base. And this is the picture I would like you to draw inside of that box. It says, draw and label a nucleotide. So I want you to draw right here. There's your phosphate, there's your sugar, and there's your nitrogenous bases. Knowing that there are 4 different nitrogenous bases that you could use. Your A's, your T's, your C's, or your G's. Alright, so I'm going to draw a nucleotide with you. So there's my circle. I got my phosphate. Okay, there's my sugar. And you should know the name of the sugar is A-deoxyribose. Okay. And then we have our nitrogenous base. So there's 4 different types, A, T, C, and G. I'm going to go ahead and choose the A. So there's my nucleotide. Alright, so I'm going to keep building onto this. And once again, a DNA is a chain of nucleotides. So here we go. Let's build another nucleotide. So come off of this sugar. And here's another phosphate. Come on down. Let's make another sugar, another deoxyribose. And come on over. And here is another nitrogenous base. So here's one DNA strand. I have, as far as the nucleotides go, we could highlight that. So we have a phosphate, a sugar, and the base. So that's one nucleotide. Let's go ahead and switch it. Get another nucleotide going here. So here's another nucleotide, a phosphate, a sugar, and a base. Alright. So here we've got our DNA double helix, and we have 2 nucleotides. So what we need to do now is make what we call the complementary DNA strand. Okay, complementary DNA strand. So basically it's just nucleotides that hook up to this. Okay. So we do need to follow the rules. And let's start down here with C. You gotta know that C, cytosine, always hooks up to the molecule guanine. So there's my G for guanine. Okay. And here is my sugar, because it's always hooked up to a deoxyribose. And here is my phosphate. Alright. So there's guanine hooked up to cytosine, because that's the base pairing rules. G always goes with C. And now we're going to come up here to adenine. And A always hooks up to T. So here comes the sugar, deoxyribose, and here comes the phosphate. So there's another nucleotide, and then we can go ahead and pair these 2 nucleotides up. So now we have a DNA double helix with 4 nucleotides, A paired up with T and C paired up with G. So you can draw this on your own paper. or in that space provided.